In 2012, CRISPR-Cas9 gave biology a tool that could find any sequence in the human genome and cut it. Within a decade, it became the foundation of modern molecular biology, won a Nobel Prize, and produced the first FDA-approved gene editing therapy. But here's what's strange. The most powerful CRISPR tools being deployed in labs right now don't cut DNA at all. They turn genes up, they turn them down, they rewrite epigenetic marks, they program gene expression like a circuit, and they're quietly redefining what genes editing even means. The CRISPR-Cas9 system has two pieces. The first is a protein called Cas9, which is a molecular scissors. The second is a short piece of RNA called guide RNA or gRNA. The guide RNA is the targeting system about 20 letters long, designed to match the DNA sequence you want to edit. Cas9 binds to the guide RNA and together they form a search complex that scans through your entire genome looking for the matching sequence. Once it finds the exact spot, Cas9 cuts the DNA. Then the DNA goes through a specific repair system which uses a provided template to precisely repair the break, allowing scientists to insert, delete, or modify specific genetic sequences. The first FDA-approved CRISPR therapy is a drug called Casgevy, which treats sickle cell disease. It works by knocking out a gene called BCL11A in blood stem cells. BCL11A normally suppresses fetal hemoglobin in adults. The hemoglobin our bodies make before birth and stop making after. Knock BCL11A out with CRISPR and fetal hemoglobin comes back online. It replaces the broken adult hemoglobin that caused sickle cell and patient gets functional red blood cells for the first time in their lives. But sometimes, cutting the DNA in a precise location doesn't solve all of our problems. For example, sometimes the level of protein's expression in the cells needs to change. The She lab at Stanford along with Jennifer Doudna's lab at Berkeley mutated the two spots that do the cutting. Just change 2 amino acids in Cas9 protein, and the results was a protein that still folds correctly, still binds to the guide RNA, and bind to the exact location in DNA. It just can't cut anymore. They called it dead Cas9, or more commonly dCas9. dCas9 is a programmable DNA binding protein. You hand it a guide RNA pointing to any gene in the genome, and dCas9 will go find that gene and physically attach itself there. It just sits on the DNA, nothing else happens, which sounds useless, but it's actually the whole point. Because the moment you have a protein that can find any sequence in the genome without damaging it, you can attach anything you want to that protein, and whatever you attach gets delivered to that specific spot in the genome. You want to turn a gene off, attach something that silences the gene. You want to turn it on, attach something that activates it. You want to change the chemical marks around it, attach an enzyme that does that. dCas9 became something biology had never had before, a programmable docking station for delivering any molecular tool to a specific location in the genome. Clinically, the field is moving fast. A company called Tune Therapeutics is in early clinical trials with a dCas9-based therapy that silences PCSK9. A gene that controls cholesterol levels. Other companies like Chroma Medicine and Epic Bio are pursuing similar approaches for liver disease, neurological disease, and several rare disorders. For 70 years, since Watson and Crick described the structure of DNA, the dream of genetic medicine has been to fix the broken letters in our genome. CRISPR-Cas9 finally made that technically possible, and is already curing diseases we thought were incurable. But the more we learn about how disease actually works, the more it looks like fixing DNA isn't the right goal for the most of it. Most diseases aren't caused by a single broken gene that needs to be rewritten. They're caused by genes being expressed at the wrong levels in the wrong cells at the wrong times. For that, you don't need scissors, you need a control panel. Now we're not just editing the genome anymore, we're learning to program it.